Today we are going to start lesson 3 of unit 3, which is molecular genetics. But before we go to today's lesson, let's recap what we have seen in the previous lesson. In the previous lesson, we have seen sex determination, especially we have taken human beings. And we have seen that humans have 46 chromosomes. These chromosomes, 23 pair comes from the male and 23 pair comes from the female. From the 23 pair, one pair is known as sex chromosome. And this sex chromosome in males is XY, whereas in females it's XX. Today, we are going to see molecular genetics. Now let's start today's lesson by an activity. What's molecular genetics then? Define what is molecular genetics. What do you understand by the term molecular genetics? Well done. Molecular genetics is a branch of genetics that investigates the chemical and physical nature of genes. Chemical as well as physical nature of genes and mechanism by which genes control development, growth, and physiology. So this is what does mean by molecular genetics. It's a branch of genetics that investigates the chemical and physical nature of genes and mechanisms by which genes control development, growth, and physiology. Now let's see chromosomes, which are a very important aspect of our topic. Chromosomes are made from two chemicals, DNA and protein. DNA, deoxyribonucleic acid, one example of nucleic acid, and histone, which is the second set of chromosome, is protein, globular protein. Whereas genes are the short section of DNA within the chromosome. In the chromosome, we do have a section of DNA within the chromosome. Chromosomes are made up of DNA and histone. The DNA part of chromosome has a gene, one section of that DNA within the chromosome. So you have to understand chromosome, gene, and how is molecule or DNA put together, molecules of DNA put together. You remember that DNA is one of the nucleic acid, which have a sugar deoxyribo, and it has also there are two types of nucleic acid, obviously, RNA and DNA. And the difference was in three aspects. One is the sugar that they have. In RNA, we do have ribose sugar, and in DNA, we do have deoxyribo sugar. And the other is nitrogenous base difference. DNA is double helix structure, whereas RNA has single strand, whereas DNA has double strand. This is discovered by Watson and Crick. DNA is made up of nucleotide, and a nucleotide is composed of sugar, phosphate, and nitrogenous base. These three, sugar, nitrogenous base, and phosphate, is known as nucleotide. So the sugar that's found in DNA is deoxyribonucleose ribose. That phosphate is found, and nitrogenous adenine, thymine, cytosine, and guanine are found in DNA. And you also remember in grade 11 biology, we have seen that pair rule. Adenine pairs with thymine and cytosine pairs with guanine. This is a base pair, and we have phosphates, and we have also sugar, pentose sugar. Pentose sugar. This is the way nitrogenous bases are complement each other. Thymine always pair with adenine, cytosine with guanine. This is the structure of nucleotide in DNA. We have phosphate group. We do have sugar, which have 5 pentose, 5 carbon. And we do have also nitrogenous base. This nitrogenous base either could be adenine, thymine, guanine, or cytosine. So this is known as nucleotide. In RNA, the sugar becomes ribose. And in the case of RNA, uracil is found. In base pairing rule, adenine always pair with thymine. Cytosine pairs with guanine. Now let's consider DNA replication. This is semi-conservative process. It involves enzymes like helicase, DNA polymerase, very important. DNA polymerase and helicase are enzymes that facilitate and speed up the chemical reaction known as replication. The ability of DNA molecule to make an exact copy of itself is known as replication. And it's base for all methods of reproduction and the base for passing genetic information. It has two uses. One is for reproduction and the other is for passing genetic information from one generation to other generation, from one generation to the next generation. Let's see cloning. The term clone is often applied to whole organism as well as to a gene. So there could be organism cloning or gene level cloning. A clone is an organism, group of organisms produced asexually from one parent. In the process of cloning, you have to remember that this is a kind of reproduction which is asexual. Only one parent is involved here. The members of the clone are genetically identical to each other and to the parent organism. This is very common in plants. So the offspring are similar to each other and to their parents. Plant cutting and cloning, and thousands of plants are produced from one parent by micropropagation, also represents a clone. So cloning, one example of cloning is micropropagation. To take a simple stem cutting, just cut off a region of a stem near to a bud. A clone of an organism is a group of organisms that are genetically identical to each other and to the organism from which they were derived. So they do have identical genetical makeup and to their parent also. Let's see genetically modified organisms. These are organisms created using genetic engineering, which contains transferred gene or genes. One gene can be transferred or many genes can be transferred, and this is known as genetically modified organisms. Organisms that are created by genetic engineering, which is very important technique and technology, which contains transfer of a gene, one gene, or many genes, and creating another organism. Genetically modified bacteria produce a range of products, including enzymes for food industry, thermostable enzymes for washing powder, human insulin, and human growth hormone. It's also important for production of vaccines for prevention of hepatitis B. This is another genetically modified, and bovine somatotrophin to increase milk yield and muscle development in cattle. Plants have also been genetically modified, and as a result of genetically modified plants, farmers and scientists have developed a disease resistant and have an improved yield. Due to genetically modified plants, disease-resistant plants have been produced and also improved. The yield is improved. And to produce a specific product, for example, golden rice is genetically modified rice, which is very important in beta carotene, important in the formation of vitamin A, which prevents night blindness. So this genetically modified genetic engineering are very important. Plants are also genetically modified, so they are disease resistant, improved yield, produce these specific products, and golden rice is as a result of genetically modified plants. The other is genetic fingerprinting. Fingerprinting is forensic technique that is used to try to solve crimes by matching DNA found at the crime scenes with the DNA suspect. So fingerprints are taken, and this is very important for investigating a criminal person. Genetic engineering that we have seen is a much quicker process than traditional selective breeding, and this looks many generations to bring about the desired improvements. A single gene transfer may achieve the same result. So genetic engineering, either single gene is transferred or many genes are transferred. What are the moral and ethical considerations using gene technology? When we use genetic engineering or gene technology, there should be ethical considerations. Genetic engineering, is it right or wrong, is a debate about rightness and wrongness of the process, because there is a question of ethics and morality. Morality is our personal sense of what is right and what's wrong, acceptable and what's not acceptable. So morality is not necessarily linked to legality, but ethics also involves a sense of right and wrongness. However, ethics are not individual opinion. They represent the code adopted by a particular group of govern its way of life. So there should be ethical consideration. This brings us to today's lesson again. We wind up here and we will meet in the next lesson.